So last week we were talking about prokaryotic cells, bacteria, but I also did an introduction to cells, so sort of laying out broad concepts. So in this lecture, we're going to take another somewhat broad concept with the cell membrane. This is a key part of all life and the cellular aspects of life. So let's start with some of the basic characteristics. We see that all cells are made up of a cell membrane, sometimes called a plasma membrane, depending on how you want to describe it. One of the four distinct features of a cell. So what we see is that if the cell is kind of enclosed entity, what actually encloses it is this cell membrane. And we'll get into the specific components here in a second. But the key thing to recognize is this is the distinct boundary that creates an interior and then an exterior within the context of the cell. And as we'll get to here, there are other aspects embedded in the membrane, such as proteins and cholesterol. Now, as far as what the membrane is composed of, it's largely composed of phospholipids. That would be the primary building component. Lipid means fat, as it says here. So what you're talking about is kind of a specific category of fat. And what makes this particular fat lipid unique is that it has dual regions of charge. From a chemistry standpoint, when we use the word polar or polarity, we're referring to something with a charge, even if it's a partial charge. So what we have is a head, which is the round part here, which is said to be charged. The contrasting part of the phospholipid are these tails. In this case, we have two tails, and they're said to be nonpolar. Nonpolar means that there's no charge. In other words, it's electrically neutral. So we have a molecule with one part with a charge and the other part with no charge. This is actually fairly unique. Most molecules are polar and or nonpolar. This one actually contains both regions of polar and nonpolar. So where that becomes relevant then is in the way it affects the characteristic of that boundary layer. So here on this next slide, we see another iteration of that first figure. What we actually see is two layers of phospholipids. So any given cell will have millions, if not billions, of phospholipids. So we don't see that quantity here, but there's very, very large numbers of these. And on the inner layer of the cell, we'll have one layer of phospholipids, and then on the outer layer, we'll have another layer of phospholipids. Now, if you'll notice, the heads will point in opposite directions. So heads will face towards the inside, and then on the other layer towards the outside. Heads are polar. Another way to describe that is hydrophilic, meaning that they're attracted to water. That would be the heads. The tails can be described as hydrophobic, being repelled by water. And ultimately what this does is it creates a scenario where water is pushed away from this internal part of the membrane. Anywhere the tails exist, water is pushed away from those tails. So what it ends up doing is it sequesters water towards the inside so it can be maintained, but also pushes the water towards the exterior. So it kind of isolates water in both regions. And that's what makes the characteristics of the phospholipids worth noting, is that they have the ability to draw water towards the head, but push water away from the tails. So that means we can maintain it both inside and outside. This is a more three-dimensional figure showing the same thing, and this is another iteration of the additional components that we'll get to next. So these terms are reiterated again here in the notes. Polar heads face outward. Polar heads are hydrophilic. So as we bring water towards the cell, it is attracted to the head. Hydrophilic means water-loving. Water, due to its nature, is attracted to that polarity of the head. In a contrasting sense, as it draws closer to the tail, it starts to push away. So it only draws near to the head, but is repelled by the tail, keeping it in a distinct boundary, either inside or outside of the cell. So that's the unique aspect of the membrane composition with phospholipids. It contrasts with the way water is a part of life in that the cell can keep it internally and also keep it distinctly externally as well. So those terms, hydrophilic, water-loving, hydrophobic, water-fearing or water-repelling, go along with the polar and non-polar nature of the tails and the heads and tails respectively. Now, if we were to build a membrane exclusively out of phospholipids and nothing else, what we would find is two things. Number one, the phospholipid layer would not be strong enough to keep itself glued together, so to speak. I'm going to pull up a video animation to better visualize that. But the phospholipids, due to their nature, want to kind of drift apart, almost like wood in the ocean, driftwood. It kind of wants to float away from each other. So what life does is it glues, if you will, those phospholipids together. And there are different strategies for this, but one of the more common strategies that you see, especially in animals and higher life forms, is cholesterol. We often think of cholesterol in terms of heart disease and cardiovascular problems. Usually it has a negative connotation to it, like cholesterol is bad. But the actual truth is, too much cholesterol is bad, and as you may know, too much of the wrong kind of cholesterol, which would be LDL cholesterol, that's even more specifically bad. So what we actually want is just the right amount of cholesterol and the right types. But putting that aside, as a sort of core component of especially animal cells, cholesterol is vitally essential to actually keep the molecules of phospholipids sort of glued together, if you will, as just a way to think about it. And I'll come back to that. Another key part are proteins that are embedded in the membrane. Think of the membrane like a wall, maybe like a drywall that encloses a room. Imagine building a room entirely out of drywall. What you'd find is that there's nothing to interact with. So what we do is we put light switches, doorways, we put interactive components into the wall to make the room more dynamic and interactive. That's essentially what proteins do. They take this sort of lifeless wall that's more like a barrier, and they allow different things to interact with it. So, for example, almost like a door, transportation proteins allow things to move in and out of the membrane, almost like a door in the wall. Communication, imagine you have a telephone or some kind of communication device, and now you can pick it up, dial in, and call someone on the outside of it. Cell recognition might be thought of like a label. You put a room tag on the outside so that you know what's going on inside that room. And cell-to-cell adhesion is a little harder to fully analogize, but this is the way that multiple rooms could be joined together. Cell-to-cell adhesion allows multiple cells to group together, which is especially important for multicellular organisms. So those are just a couple different functions that we'll come back to in more detail. Now, the membrane, as I mentioned, wants to kind of drift apart, and this is described as membrane fluidity. And I'm going to pull up that video here in the next section, in the next part, to visualize that better for you. But we often describe it as semi-fluid. Unlike building material in our world, which is solid, building material in this nature at the life level is sort of semi-solid, semi-fluid. So the phospholipids kind of drift and float around each other, and that's where that term comes from. It allows them to remain in close association. It allows the membrane to kind of change shape, form itself, and create a more dynamic equilibrium in reference to the environment around it. So imagine a room that could sort of shift and morph in reference to what's happening around it. That would be kind of like the nature of this fluidity here. So let's take a real quick second, and I'm going to pull up a video on YouTube. So this video comes from the channel Structure of the Cell, or excuse me, the title of the video, Structure of the Cell Membrane, from the channel Nucleus Biology. I want to give them credit for this video here. I'm not going to play the whole video, and I'm going to mute the narrator, but I just want to give you a chance to visualize this real briefly here. So starting kind of in the middle, if we're looking at the outside of a cell from an animated standpoint, we would see a top layer and a bottom layer made of phospholipids. Here's the heads on each side, tails facing back in the middle, and the animation kind of lets you see the fact that yes, it is sort of fluid, moving around. It's not a static, solid kind of configuration. Up here in the background, we can see these proteins. Now it's going to zoom in on the phospholipids, to reiterate what I kind of already talked about, that you have hydrophilic heads is what it's talking about here. Let's go forward just a little bit. Now it's showing the hydrophobic tails embedded in the middle. Those are going to ultimately repel water out of that space, either direction, in terms of the inside or the outside of the cell. Now, proteins, which we'll get to in more detail, these are now embedded in the membrane, and kind of like the phospholipids themselves, they actually float around. So this is what's partly referred to as the fluid mosaic model, which I'll bring up here in a minute. So the fluidity is in reference to the phospholipids, and the mosaic is in reference to the pattern that the proteins exhibit if you're looking at them kind of from the exterior side of the cell. So that kind of gives you a better idea of the fluidity of it, and we'll go ahead and go back to the lecture notes here. Membranes are in constant motion, and one of the things that affects the level of motion is temperature. What temperature ultimately does is it kind of speeds up molecules at the molecular level. So a higher temperature is actually going to cause those phospholipid molecules to move around at a higher speed, which is true for all molecules. The higher the temperature, the more speed molecules are moving around at the molecular level. It's ultimately why temperatures have to be balanced. If you get too much heat, you'll get too much motion. It becomes too chaotic. You can't keep it all together. And in contrast, if you have too low of a temperature, you don't have enough motion, and everything kind of becomes too solidified in a way that's not compatible with life. So temperature, we all know temperature affects us. One of the reasons why, not the only reason, that temperature affects us is because it actually has a direct impact on the fluidity of our membranes. So too hot, too much movement, and what technically happens is the membranes can become leaky. One of the reasons why you can't sustain life at too high a temperature is the membranes start to actually fall apart and leak. And then at the other end of the spectrum, at a too low of a temperature, that movement solidifies down to a slow crawl, and the necessary fluidity is lost, and membrane proteins will typically either reduce or stop functioning at these low temperatures. Every life has a different temperature range, but generally speaking, that's kind of the nature of what happens at too high or too low. Bring back cholesterol here. This is one of the things that cholesterol does. Not only, as I mentioned, it helps kind of glue the membrane together, but it also helps resist those temperature changes. So what happens with cholesterol is as the temperature goes up, cholesterol, it acts like a buffer to resist that leakiness that will come along at some point. So at higher temperatures, cholesterol pulls that fluidity back towards the equilibrium and keeps it from becoming too leaky. At lower temperatures, the cholesterol does the opposite effect, where it sort of brings it back up into a normal level of fluidity. So you can think of cholesterol kind of like a buffer in this regard. It keeps the temperature range, it keeps the movement of the phospholipids more in the right range, even as the temperature goes too high or too low. Now there's still a maximum range, so even with cholesterol you can still go too high or too low, but it does maintain a little wider range of fluid equilibrium due to the presence of cholesterol. So that's one of the things that cholesterol does there. Now another concept is the nature of the fats themselves. So you've probably heard the terms saturated and unsaturated fats. So when we start building phospholipid tails, we can have a combination of both saturated and unsaturated fats in the tails themselves, which is a sort of more in-depth conversation about that. But the tails themselves can consist of both saturated and unsaturated types of characteristics. So what you'll find is that unsaturated fats tend to have more kinks in their tails, and this is due to the fact that they have double bonds, more technically speaking. Double bonds ultimately cause the tail to kind of bend and kink, and what that does from a technical standpoint is it forces more space, as you can see here in this upper left. It forces there to be more space naturally between the phospholipids. Saturated fats, on the other hand, contain straight tails. The saturated nature of a tail means that it doesn't contain those kinks or those little bends. And so as a result, the membrane will stay closer together as is generally intended. So the nature of the two types of fats brings the sort of equilibrium into balance. So typically you're going to build more with the saturated fatty acid tail so that you have a tighter membrane surface as opposed to a leakier, more dispersed membrane surface. Another distinction is the length of the tail. So there is a way to increase or decrease the length of that tail. So longer tails will generally cause the membrane to become less fluid. Shorter tails will tend to make that membrane more fluid and more capable of moving about. So those are some differences that do occur throughout life. Generally, different species are more consistent in the nature of their saturated, unsaturated, length of tail, short, long, and also the temperature preferences that they tend to live at. So what we see is that life tends to adapt its membrane in reference to the environment, and that different life forms have those characteristics based on where you typically find them. So when you look at temperature, one of the things that life does is it changes cholesterol depending upon its temperature conditions. This is something of a epigenetic response, meaning that the cell can increase or decrease cholesterol based upon its temperature. So the range of adaptation is limited, but there is a certain amount of wiggle room, if you will, so that an organism can sort of migrate one way or the other its cholesterol levels to slightly move to a higher threshold of temperature or a lower threshold of temperature. So this is noted in certain types of fish, especially fish that live in cold temperatures. So they're able to change that cholesterol in order to kind of fit the temperature that they're living at. Another adaptation is seen in plants. Plants can change the length of their tails to do the same thing. So they can actually make it more suitable for a hot climate as opposed to a cold climate, where changing the length of the tail will actually give them a slight advantage if they're found in too hot of an environment. And this is one of the many things that accounts for why certain organisms can live at a higher temperature where others can't. If you've done any gardening or if you know much about plants, you'll know that some plants are pretty well off in a hot temperature and other plants don't do well at all in a hot temperature. So these are just some of the many things that plants can do at the membrane level to change that sort of favorability at different temperatures. Same thing with fish as the other example I gave here. Now, I mentioned the fluid mosaic model. This is a picture of a mosaic. It's a type of artwork you're probably familiar with, where you build an artwork out of little patches. And ultimately, you get kind of a broader picture, one little part at a time. That analogy or metaphor is used to describe the cell in terms of the mosaic. What you see is on the surface of the cell a similar pattern that, at least in somebody's opinion, kind of matches that mosaic pattern. So that's where that term ultimately comes from. So somewhat of an arbitrary concept, but the term fluid goes back to the phospholipid movement, and then the mosaic is describing this kind of patchy pattern that you would notice if you looked at the cell membrane in great detail. That those mosaic patterns are usually characterized by the different proteins that stick out on one side or the other of the membrane, as well as some other components like the carbohydrates and cholesterols. So you get this fluid mosaic term in reference to the pattern and the fluidity of the phospholipids. Now, here we're going to talk more about these membrane protein functions. You can't have a membrane without having proteins in terms of life as we know it. So the phospholipid is one part, the protein components are another part. So one of the things that the proteins will do in no particular order is they will help fasten one cell to the next. So here you can kind of see a couple different examples where one protein sticking out from one membrane attaches to another protein on a separate cell, and it helps sort of stick them together. Those are adhesion proteins. There's many different types, but that's a broad kind of category. Another, as I mentioned, are identity tags. These are called recognition proteins. So this is important in a number of different aspects of life, but self versus non-self is a way that we describe cell recognition patterns in the immune system, which will come up later in the semester. So your body has to recognize its own cells and distinguish those from foreign cells. That's kind of how the fundamentals of the immune system work. So self would be recognizing your own receptors or your own protein patterns. Non-self would be recognizing foreign protein patterns, and the body can distinguish between the two based upon recognition proteins. By the way, that's also how organ rejection works. If you try to do an organ transplant, the body of the recipient will recognize the pattern of the organ as foreign and attempt to reject it. And that's why you have to do anti-rejection drugs or immunosuppressants when you do organ transplants. Another is a receptor protein. So I mentioned in the introduction to cells that cells communicate. So they do this, at least in one way, with receptor proteins. So you'll have a protein on the outside that binds to a signaling molecule that causes some kind of reaction on the inside. So it's almost like the way you would recognize somebody's voice or you would visualize somebody's appearance. You have a recognition that is recepted by your senses, and then it changes, theoretically, your response. So if you see someone you're familiar with, you respond one way. If you see someone you're unfamiliar with, you respond a different way. Receptor proteins make up this highly complex nature of the way cells respond to their environment. But in simple, it's a protein on the outside of the cell that responds to signals that changes the way the cell behaves in different feedback mechanisms. So examples would be the way your body metabolizes and grows. It gets signals that change its metabolic patterns and growth patterns. It also relates to things like movement and response. So if you get a positive signal, you might move towards that signal like a nutrient, or if you get a negative signal, you might move away from it like a toxin or a threat of some kind. Another example, very common, are transport proteins, which are probably most comparable to something like the door in the wall, as I mentioned earlier. So you wouldn't build a room with no entrance or exit. The same is true for cells. You're going to build little entry-exit points that act in this case, that transport proteins act in this case like those gates or doorways that move things back and forth. The specific examples are highly complex, so every life form and even different cells within your own body will all have their own unique transport proteins, but broadly speaking, they're universal across life. So this is how you get certain nutrients into the cell and expel waste out of the cell. That goes back to the idea of homeostasis, which I mentioned in the introduction to cells. All life has to exhibit homeostasis, bring nutrients in, expel waste at a minimum. So those are kind of key aspects to how you do that. So electrolyte concentrations, pumping in ions, electrolytes, sodium, potassium, those types of things are done through specific transport proteins. And you probably, if you've taken anatomy and physiology, can think back to some examples of like sodium, potassium pumps and things like that. So those are more specific examples of transport proteins. So transportation is going to involve a protein that moves all the way across the cell. These are sometimes called transmembrane proteins. They move, or they exist on both sides of the cell, and so therefore any molecule can go all the way in or all the way out across that membrane. These are usually selective. So kind of like a door, but imagine a door with a highly specific key card or a lock that you have to have a specific key to get through. So it's not like an open door with a broad invitation. Typically, it's more of a selective door that says, hey, you have to have this exact password or key card to get in. That's the idea of selectivity. Receptor proteins have similar configurations. The exterior will have a configuration like a key and a lock where it will also require a specific binding molecule. So this is where the complexity of receptor proteins gets to be fairly nuanced. You can have theoretically thousands of different specific kinds, and kind of like the transport proteins, only certain key cards will open those or bind to those receptors in this case. So for example, when it comes to hormones, you'll have specific receptors that only bind hormones like testosterone or estrogen or cortisol, for example. Neurotransmitters, these are found on your neurons. You'll have receptors that only have a binding site for dopamine or serotonin or acetylcholine. So each receptor binds a specific molecule, and that is sort of like a specific lock and key interaction or a certain key card kind of interaction. Another example that applies to the immune system are cytokines. Cytokines are immune signaling molecules that the immune system uses to change the immune response based on what kind of situation you're in. Adhesion proteins facilitate cell-to-cell attachment. They'll allow two cells to bind together. This also goes along with what's called the extracellular matrix, which is something that sort of lays exterior to the membrane, a series of carbohydrates and proteins that also help glue cells together. Extracellular matrix is something that you see very heavily in, say, bone tissue, for example. So bone almost looks like a rock kind of material. And what you'll actually find is that the cells are buried deep inside the bone, and what the bone is largely made up is that sort of calcified material, almost like rock. That's actually the extracellular material. So bone is kind of an extreme example where the cells are buried deep inside the extracellular matrix, and in the case of bone, that matrix becomes calcified, almost like a rocky mineral, which is largely extracellular. So every cell does that differently. Cartilage and joints do a similar thing. So the cell is kind of buried deep within the matrix, which is an exterior that kind of holds all those cells together. So that's another concept with extracellular matrix, but that also helps explain how cells glue themselves together. So adhesion proteins kind of facilitate the binding of the matrix to the cell, and then the matrix goes on to take a more complex adhesion property. Recognition proteins, these are often glycoproteins. So a glycoprotein is a protein with a complex sugar attached to it. So very often when we're recognizing a cell, self versus non-self, we're recognizing the specific pattern of protein and carbohydrate. And this is one of the few places that carbs are used in the body that's not for energy. Most of the time carbs in animals in particular are used very, very heavily or primarily for energy, like sugar. But in these types of situations, they also serve for that identity pattern. The specific recognition that we get is based upon the combination of proteins and carbs. Now, in terms of the way these membranes are positioned, I mentioned the term transmembrane. Transmembrane goes across from one side to the other, so these are good for acting like a gate or door in the cell. Integral proteins are embedded on one side or the other. So these will be good, for example, picking up a receptor signaling molecule on the outside or responding to a different signal on the inside. So either way, they're on one side or the other, either inside or outside. So trans, across, integral on one side or the other. A couple of examples to relate these to the human body. Recognition proteins would be, an example of that would be red blood cell markers. So you might think of your own blood type. Your blood type will either be A, B, AB, or O. There's actually only two recognition types, which are type A and type B. So generally, you'll have one or the other. That's what's most common. You'll have type A blood or type A or type B blood. In that case, you express only one recognition pattern or the other. If you have a more rare blood type, it's possible to express both A and B, in which case you're type AB. And if you're type O, what that means is that you have a condition in your genetics that cause you to express neither. So with type O, you don't actually have a type O receptor. You lack either the A or B, and therefore you're characterized in the absence of an AB receptor, A or B receptor. Another example is vasopressin. So vasopressin is a peptide hormone, and this hormone ultimately causes your body to change the way it retains water. So it's a signaling hormone that sort of gives you a feedback as to whether you have too much or too little hormone. In this case, too little, and so it will cause the body to hold on to water. And so the signal occurs on the outside of the cell. giving the body an indicator that you're probably dehydrated or stressed, and so you hang on to water in response to vasopressin. Last thing here is not all proteins fit into one category or another. So an example would be integrins. So integrins are an example that play both a role in cell adhesion and as a receptor protein. So they can connect cells together and also act as a receptor to transmit signals from one side or the other. So that's just kind of an example to where not every type fits exactly into one category or another. So those are just a few examples that you would find in the human body. All right, that's the end of this lecture. Next week we'll be talking about viruses. So we'll go into a different concept with pathogens that are most common to humans being viruses.